close to a vaccine for breast cancer. And I had a conversation with Dr. Nora Disis from the University of Washington's Cancer Vaccine Institute, and my mind was just blown. And so we've been talking and are actually going to make a collaborative to accelerate a breast cancer vaccine. And we'll be talking a little bit about that later after she presents. And it's going to be an effort that we really want everybody to be involved in, because this impacts all of us, both preventing recurrence, preventative for all of our loved ones, so that nobody else needs to go through what we all have been through with this disease. And so I'm really excited for all of you to see her presentation and to learn about the process of a cancer vaccine, a breast cancer vaccine, and how close we actually are. So with that, I'm going to turn it over to Dr. Nora Disis. She is the director of University of Washington's Cancer Vaccine Institute, and considered by many to be the nation's foremost expert on breast cancer vaccines. She's already got some in trial. And so with that, Dr. Disis, welcome.
Thank you very much, Kristen. I'm going to share my slides with you, and I hope everyone can see those. If you have any questions during the talk, feel free to write them in the chat or raise your hand. We can take a quick break and answer them. Today, I'm going to talk about vaccines to treat and prevent breast cancer. And this is something that I've been working on for the last couple of decades. And I just want to let you know about progress that we've been making, and the fact that there probably will be a vaccine to prevent breast cancer recurrence within the next 5 years or so. You know, when you think about it, vaccines really have had the most profound impact on human health than anything that's been developed. Diseases that used to kill us just 2 generations ago, like diphtheria, flu, pneumonia, measles, mumps, can be completely prevented as long as people would get their vaccines. And in fact, you can see on this slide that we've been able to decrease the amount of disease in some cases by 100% or 99%. We don't even hear about these diseases unless there's an enclave of people who are against vaccination. So you would think that with all the technology of creating infectious disease vaccines, that cancer vaccines should be a reality by now. And in fact, cancer vaccines started study about as early as the first infectious vaccine for smallpox. But there have been a lot of problems in terms of being able to bring forward a cancer vaccine. Although I'll have to say, within the last decade, we've really learned a number of things that have allowed us to make cancer vaccines, especially breast cancer vaccines, a reality. What we're trying to do with a vaccine is to ignite the immune system. Our immune system defends the body against infections and lots of other threats while protecting the body's own cells. And that's the beauty of the immune system. It's very, very specific. Our goal has always been to use vaccines to train the immune system to recognize cancer as a dangerous threat. In some cancers, that is a little easier than other cancers, because some cancers have a lot of mutations or are caused by viruses. And those two things are already inherently foreign to the immune system. But for the most common types of cancers, like breast cancer, ovarian cancer, colon cancer, prostate cancer, these cancers are not associated with a lot of mutations, and they're not associated with a virus. So our group has been developing vaccines to try to treat and prevent these most common types of cancers. And I can say we are at a tipping point for cancer vaccines because of 3 major things, and I'll get into these in a little bit of detail. The first thing is, we know the type of immune response we need to kill cancer, and it's not the same type of immune response we need to stop an infection. For infections like COVID or the flu, we need to generate antibodies. But for cancer, where the immunogenic proteins are really hidden inside the cell, we need to ignite the arm of the immune system called the T cell. So that was a big understanding that came about within the last decade. The second big tipping point is now we know what parts of the cancer can stimulate the immune system. So if you develop a vaccine, you need to know the immunogenic proteins that you would encode in a vaccine or put into a vaccine. And now we know literally hundreds of immunogenic proteins in breast cancer. The trick is just picking the right ones. So we know what antigens or immunogenic proteins can be targeted for breast cancer. And finally, we have new vaccine technologies that make immunization much more effective. And while there's a lot of vaccine technologies out there, the ones that we focus on are nucleic acid vaccines. That's either DNA or mRNA. Now, why are those more effective? Because DNA and mRNA allow your own immune system to get involved in the vaccine so that when you get immunized, the DNA or RNA actually goes into a skin cell and allows your immune system to process the antigens and present it to your immune system the way your own genetics dictate. Also, RNA and DNA vaccines are very inexpensive. They literally can be made for $30 or $40 in immunization, and that allows everyone to be able to be vaccinated. And finally, RNA and DNA vaccines have a long history. They've been used for a long time, even though we only have become aware of them with COVID, and they are really, really safe compared to other vaccine technologies that are out there. So I said that one of the biggest tipping points is we now know what type of immune response is needed to eradicate cancer. And in general, when you talk about T cells, and these are living cells that can stick in our body literally forever and confer immunologic memory so that we always have T cells circulating, capable of recognizing whatever antigen we train it to recognize. There are two flavors of those T cells. The first flavor, the one we really need, is called a type 1 response. And these are CD8 T cells, which are killer T cells, and CD4 T cells, which secrete substances to really inflame the environment. And these two types of T cells together, type 1 T cells, generate the immune response we need to kill cancer, almost like autoimmunity at the site of the cancer. The type 2 immune response is what we generate to kill an infection. You get B cells and regulatory T cells, which absolutely shut down the type 1 response and tell the body you need to make lots of antibodies. And unfortunately, for common solid tumors, this is the most common immune response we see. So I can take blood from any patient with cancer, and only in a few patients will I find a type 1 immune response. But in most patients, I can find dozens and dozens of antibodies directed against antigens expressed in their cancer. But the antibodies can't kill the cancer. Only the type 1 T cells can. So the first key in an effective vaccine is eliciting the right type of immune response. Well, why is it? Why is it that our bodies want to create that antibody response against cancer? The most important reason is that many of the immunogenic proteins that are expressed by breast cancer don't appear dangerous to the immune system. They're normal self proteins that have gone awry are highly upregulated, are causing cancer to grow, but they're not different enough from self that the immune system creates the attack response. So what happens is the immune system recognizes that something's not quite right with those proteins, but creates the type of immune response that doesn't kill them, but tries to heal them. Whereas if you have a disease like cervical cancer that's caused by HPV, or you have a tumor that's highly mutated like melanoma, the virus and the mutations can give that danger signal and be seen as foreign by the immune system. And these are the types of cancer patients that are most likely to have more of a type 1 immune response just naturally to their tumors. So all cancer vaccines have to be designed somehow to switch that type 2 immune response to a type 1 response to generate that type 1 T cell response. Now, I said we have safe and effective vaccine technologies, and I just want to go over again how DNA and mRNA technologies work. You take these nucleic acids, and here's an example of a vaccine that you'd get in your arm like any other vaccine. And this is kind of how the COVID vaccine worked. The way that you present antigens to the immune system is really dictated by your own immune system genetics. And so if someone gives you a fragment of a protein and your body can't process that fragment, you won't make an immune response to it. By taking DNA or mRNA and injecting it into your skin, those nucleic acids are actually taken up by your skin cells, and for a very short period of time, the immunogen or antigen is made inside your own cell. And your own immune system machinery will chop up and present fragments of those antigens to your immune system in the way your immune system most wants to see it. And that is why these are more effective in generating immune responses than, let's say, a protein vaccine or even a inactivated virus vaccine, which most of our childhood vaccinations are those two types of vaccines. So if we go into clinical trials with technology that generates those, most likely generates those type 1 T cell responses using these safe and effective vaccine technologies, it's really a whole new world for breast cancer vaccines. So how do we do this? Our technologies, again, are really designed to target those non-mutated proteins that are antigens. So the first key is, out of all those hundreds of antigens, which ones do we choose to put into a vaccine? So we use genomic technologies to identify proteins that are the cancer drivers. And I'm going to give you a good example of one in the next couple of slides. Then we take that protein, we break it apart, and we use artificial intelligence to indicate to us which are the segments most likely to interact with the immune system. Then we create those segments chemically and perform high throughput functional screening to show which of those segments preferentially will elicit that type 1 immune response we need for cancer eradication. This gives us a barcode for an effective breast cancer vaccine. Then we literally encode that vaccine into DNA, make that DNA, and use that DNA as a vaccine. We're developing DNA vaccines to treat cancer. We give them to patients along with immune checkpoint inhibitors or other immune therapies in breast cancer patients who have advanced stage disease and are not responding to other therapies. But we really focus on preventing the disease from coming back by immunizing patients once they've finished treatment. And we even have vaccines that we're developing to prevent breast cancer from even occurring in the first place. And I want to show you examples of these latter two approaches. So one of the first vaccines we made targeted the HER2/neu protein. Now, many of you might be familiar with HER2/neu. It's a non-mutated protein that becomes very highly upregulated in about 30% of breast cancers. And it's a driver of that type of breast cancer. In fact, it's so important, we even call that type of breast cancer HER2/neu positive breast cancer. So here's a normal cell that has low levels of HER2 and a cancer cell where you may have 100 times or 300 times more the amount of HER2 that you have in normal cells. And it's that marked overexpression that allows the immune system to be very selective to the cancer. The question is, how do you make that response be a type 1 immune response instead of a type 2 immune response? So we took the whole HER2/neu protein, we ran it through our artificial intelligence programs, we did high throughput functional screening, and we found that the intracellular domain of the protein held all the type 1 selective components of HER2. So if we removed most of the HER2/neu protein and just immunized with the intracellular domain, in animal models, we were able to get very high levels of type 1 T cells and a potent anti-tumor response, and it was safe. So we did a phase 1 study, tested 3 doses of the vaccine in a lot of patients, in 66 patients, 22 patients per arm. And we looked at advanced stage HER2/neu positive patients from stage 3 and stage 4. We gave the vaccine, just like a COVID vaccine, 3 times about a month apart. And this study was actually done more than 10 years ago. It was the first DNA vaccine used in cancer patients, and the FDA required that we had a very long follow-up for the patients to make sure that there were no untoward effects. I'll get right down to the brass tacks. The HER2/neu ICD vaccine elicited a very strong immune response, and it appears to be effective. All 3 vaccine doses tested elicited high levels of HER2/neu specific type 1 T cells. The middle dose, which was the 100 microgram dose, this is in red, was really the most effective, not only from generating the highest magnitude of responses that lasted over a year after vaccination, but 80% of the patients with advanced stage breast cancer are still alive over 10 years from the end of vaccination. Even with the less immunogenic doses, we still have 70% of advanced stage breast cancer patients alive 10 years after vaccination, and 50% of the patients have not recurred their disease. So this vaccine was picked up by a company called Aston Sci, and right now it's in a randomized phase 2 study that hopefully will lead to a randomized phase 3 study to be able to potentially have a vaccine for all HER2/neu positive patients to be able to prevent disease recurring. And there are many other clinical trials that are being planned by investigators across the United States with this vaccine. One thing about our institute is, we can't do it all. So if you've got a great idea and we have vaccine, you can have the vaccine to do whatever you want in terms of taking it into different patient populations. But it's very unusual to find a vaccine like the HER2/neu ICD vaccine, where there's a very strong biologic driver for HER2. So what would be another concept? And so one of the vaccines we've developed is a vaccine targeting cancer stem cells. And this vaccine is actually aimed to prevent breast cancer, and we call it StemVac because it's targeting cancer stem cells. Now, why the cancer stem cell? We have stem cells in our body. We have to have stem cells in our body. Any cell that needs renewal has a stem cell attached to it. But in some cases, especially when you're older, because cancer is generally a disease of older people, 50 years old, 60 years old, over your lifetime, you develop genetic hits for whatever reason. And your cancer stem cells, your normal stem cells, in some cases, aren't able to correct the abnormalities that occur. And then you develop a stem cell that becomes neoplastic. And these now cancer stem cells in tissues, mammary tissue being one of them, begin to become immortalized, and they are resistant to therapy. They're the ones responsible for recurrence, and they become metastatic very early. When you talk about circulating tumor cells, you shouldn't have circulating tumor cells. Once your tumor cells become circulating, that means they've lost the ability to want to be with each other. And that's kind of the beginning of metastases. That's why chemotherapy can kill those cells once they get into the circulation. But it's an early indicator that in many breast cancers, you already have those early cancer stem cells. We looked at proteins that were involved in driving the cancer stem cell. And we chose 5 proteins across multiple different cancer stem cell pathways. And we took those proteins and used our rubric to identify portions of the proteins that could generate that type 1 immune response. Then we created a multi-antigen vaccine, StemVac, and we began testing it in mice. This is an example just showing you the power of a vaccine like this. These are transgenic mice who were genetically engineered to 100% develop breast cancer at about age of 150 days, and most of the animals are dead by almost a year of breast cancer and metastatic breast cancer. When we began to immunize animals at about teenage years, we could prevent the development of breast cancer in 50% of those mice. In fact, they're well over a year out, and they've been breast cancer free for over 100 days. In the animals that still developed cancer, their cancer remained localized and didn't metastasize and could be removed with surgery alone. The animals were safe. They didn't develop any evidence of autoimmunity. So we took StemVac into a phase 1 dose escalation study. Again, 3 doses of vaccine. We looked at advanced stage breast cancer, stage 3 and stage 4, not HER2-neu. This time we looked at hormone receptor positive and triple negative breast cancer patients. We looked at 10 patients per dose, 30 patients in all. We gave the vaccine 3 months intradermally, 1 month apart. But this time, like the COVID vaccine, we gave boosters at 3 and 9 months. Again, the intermediate dose was the most immunogenic dose. And here you can see no one had an immune response before vaccination. After the vaccine, about half the people had an immune response. And after the booster shots, all but 1 patient had an immune response. And when you look at those responders, they were able to boost their immunity to the StemVac antigens 500-fold over what their baseline response was. We could detect an immune response to all 5 of the stem cell proteins. The boosters really increased the level of response, and we generated very high levels of StemVac specific immunity. 1 in 300 of their white blood cells was a type 1 T cell that recognized cancer stem cells. So STEMVAC is aimed for cancer prevention. We have a clinical trial right now in triple negative breast cancer where we're immunize patients to try to prevent disease recurrence, which is a major problem in patients with triple negative. But also the BRCA1 gene mutation is associated with creating cancer stem cells very early. And so we hope to get a trial started giving the vaccine to BRCA1 and 2 gene mutation carriers to try to prevent the development of breast and ovarian cancer. But as you can see, there are many diseases that are highly associated with cancer stem cells. So this also gives you an idea of a vaccine that may be able to have broad applications. So I'd like to end the last slide with talking about the biggest heroes in our work, which are our patients, because we would never be able to go forward without having patients who agree to volunteer in these vaccine trials. And I can tell you, many physicians don't believe in cancer vaccines. So it's not like chemotherapy where physicians are always ready to put a patient on a chemotherapy trial. So many of the patients who join our clinical trials find us, and it's really with them that we can create a path to ending cancer as we know it. This is a person who participated in our DNA vaccine trial. And she sent us a letter about 2 years ago and said, I participated in the HER2/neu vaccine trials about 5 years ago. Just to let you know that I'm doing great. A lot of living in these past 5 years. Both parents have passed and both my children have gotten married. 3 grandsons born, twins too, as you can see. I think often of the trials and how grateful I am that I qualified and was able to benefit so much from the program. Her disease was stage 4 disease, and being able to maybe have participated in extending her life is one of the greatest privileges we have as scientists. So I'd like to acknowledge my group, the UW-Medicine Cancer Vaccine Institute. I'm a mouthpiece. It takes a lot of people to be able to do work like this. A clinical team that develops all the protocols and runs all the clinical trials. A translational research team, which develops the vaccines. Experts in mouse modeling, which helps us generate the data needed to go to the FDA. And our immune monitoring laboratory that helps us run multi-center clinical trials really all across the United States of our vaccines and helps us figure out if they're generating the types of immune responses we're looking for. And with that, can I answer any questions for you?
I'll ask the first question, if that's okay. As people maybe log in their question in the Q&A in the bottom. As you know, Dr. Disis, I have metastatic breast cancer. I was diagnosed 7 years ago, and this summer marked 5 years of being having no evidence of disease detected in my body, and I was allowed to go off chemotherapy after 7 years, which feels amazing. How does someone like myself reach out to you or your team, your clinical team, to volunteer to be part of this?
The ability to volunteer really depends on whether we have an open clinical trial that matches your criteria, right? What we would love to do is have like an ongoing open study, but there's no one really that funds that type of thing because it's really an ongoing long trial. So the type of vaccine you would qualify for would be like a first-in-man vaccine because you're the type of person, we say if you've had stage 3 and stage 4, you're in a remission or have bone-only disease, this gives us the time to immunize you. We have a website. It's www.uwcvi.org, where we open all the clinical trials we have ongoing at UW, University of Washington in Seattle, of our new vaccines, but we also post all the vaccine trials we're participating in. And that includes when we give our vaccine to other people to run their clinical trials. We always say the payment for that is we're a site, meaning we'll be a site and enroll patients in that trial you're developing. So there's lots of clinical trials listed there. And then you can always just keep in contact with me directly.
And sorry, just to follow up to that, is that are those vaccines strictly for the triple negative for early stage, and then as well the HER2 positive?
Currently, the HER2 new vaccine belongs to a company now called Aston Sci. So they are going to do the commercial development of the vaccine. And they have some studies going on in HER2 new positive cancer and also HER2 new low breast cancer. There's a trial I know that is being developed by a woman named Paula Pullman at MD Anderson and one of our physicians in the UW CVI named Shweta Vinayak. And that is going to be the follow-on trial to the one I just presented. That will be in advanced stage cancer patients. They've developed it. They're looking for funding for it right now. I think they're going to put in a grant to the Department of Defense and the National Cancer Institute this spring. So that one would be about a year away.
Thank you.
And Jillian, this might be a good time as we're waiting for more questions to talk about how Dr. Disis and you and I and some other survivors are working on putting together a nonprofit coalition to bring all of these researchers together to help accelerate this in a safe way. If everybody's working together, as Dr. Disis explained it to me, everyone at this point is working in their own silos. And it's not that they don't want to cooperate, it's just a massive task. They're running their trials, they're very busy. But we're working on putting together a coalition of scientists, of survivors, of people who can help with the fundraising and financing. And our hope is that we bring all of these pieces together. We work with the government, with the FDA, with the NIH, hopefully, and we're able to really bring these trials, make them bigger, more robust, and in that way, help push them through sooner, so that Dr. Disis said 5 years. I'm hopeful that maybe we can even do it before that. And so we'll be keeping this community abreast of where things stand with that and how everybody can help out. But if everybody is working together on these vaccines and doing these trials, I really think that we could have something soon.
So we have, excuse me, we have a couple questions in the chat box. Rebecca asks, are there any vaccines on the horizon for ER/PR positive HER2 negative breast cancer? It's also the kind of breast cancer that I have.
We have two vaccines. I mean, one of them is StemVac. The ER/PR positive disease is also associated with cancer stem cells. And we think that this would be a good vaccine going forward for hormone receptor positive disease. We also have another vaccine. This vaccine also is with Aston Sci that targets 3 proteins. And 2 of those are associated with the insulin-like growth factor receptor family, which is very operative as a driving force in hormone receptor positive breast cancer. So those 2 are working their way into phase 2 clinical trials.
Okay, there's another question. Hi, Maureen. I'm so glad to see you're here listening. So she says, incredible work, so important. She missed the very beginning of this, but she asked, if someone is triple negative and 10 years out from stage 2 breast cancer, can you get the vaccine once it becomes available, or only with current higher stages?
I would probably say when the vaccine becomes available, now that means it's been approved by the FDA and there's still a while to go, you would be able to probably immunize anyone after they've had breast cancer. It's not like a virus where if you've already got the virus, you probably can't be immunized. If you have triple negative breast cancer 10 years ago, the vaccine is to immunize you in case it would ever come up again. Your immune system would be primed to eradicate it before, the cells before you even knew you had it. So I envision that once the vaccines become approved, it doesn't matter how far out you were from your breast cancer, you would be able to get the vaccine. Right now, the stage that the vaccines are in, they're in the stage of trying to prove that they are clinically effective. And that's still in the pre-approval stage. And it's the type of clinical trial that is super specific. So usually what happens is the patients have to be all in this one area of treatment and then get the vaccine and move forward. So right now the vaccine isn't broadly available. It's only available in the context of these very specific clinical trials.
So just a follow up to that, Dr. Disis, in your slides, you were referring to mice that received that were intentionally bred to have breast cancer. And then when they were sort of in their teenage years, for lack of better comparison, they were given the vaccine and then they were allowed to live much longer. Is the idea that, like vaccines for HPV to prevent cervical cancer in women long term, is that sort of like a comparison that we would eventually see our tweens getting the vaccine at a particular age?
I don't see tweens getting the vaccine because breast cancer is really caused by these non-mutated tumor antigens. So they're not really specific like HPV is a virus. So you really need like long-term safety data. I think where I see the vaccines are you're premenopausal, okay? So in your, early 40s, once you're done with child rearing, you would start your midlife vaccine series. And that's when you'd get the preventative vaccines. So I don't see giving them to kids or giving them to tweens or even in the 20s unless you're at genetic high risk. For the regular population, I see this as kind of a perimenopausal type of vaccine series.
And that's interesting, because our community, our I Rise Above community, consists of women in their 20s, 30s, and 40s who are quite honestly a far away from being menopausal, and
Yeah.
so because we have to go on all of these types of medications, that then we're kind of thrust into that space.
Yeah, you guys would be considered higher risk. So with high risk women, and I do have to say we're defining better breast cancer risk all the time. I see a world within the next 10 years of women coming into their primary care physician and answering a questionnaire and getting a genetic test with a panel of hundreds of genetic, mutations, where we now have highly quantitated which ones really confer risk, and you're given a risk category. And how we would follow you throughout your lives, starting at age 20, let's say, would be hey, if you were at very high risk, you'd probably get the vaccine early. If you were at intermediate risk, you might start mammograms at 30. You know, the risk benefit ratio would be, we'd have paradigms of what we would do that would best follow your risk.
So I'm just sitting here being the devil's advocate because I hear so many stories of young women who were not genetically predisposed. They didn't have any family. They wouldn't have been, I guess, categorized as being high risk, including myself. And so what do we say to those women who are just, are interested in maybe getting the vaccine, if that is ever a possibility?
I would have to say we'd have to have a lot more safety data if women wanted to still have children. You know, the last thing you want to do is give a vaccine and have an untoward effect against a child, right? Because your immune system can get across the placental barrier. So I really see the vaccine as, until we have very long-term safety data, meaning, you know, some of our vaccines that we're giving to breast cancer patients, and then, you know, they start menstruating again, and then they decide they're going to have kids. And this data is being collected. We do have women who've had children after vaccination. We have to get a lot more of that before we go into widespread, you know, if you haven't had your kids yet, it's fine to take the vaccine. You won't have a problem, or your children won't have a problem. That's my biggest worry about just, you know, giving the vaccine to everyone. If you're below the age of 40 and you've developed breast cancer, you've got a genetic risk. We just haven't found the gene. And one of the things that is happening right now in multiple labs across the United States is identifying these pathologic variants that we've never really identified before. You know, I'm sure you guys are aware there are so many programs where you can volunteer as someone who's had breast cancer or has a family history of breast cancer to donate blood so that people can find these variants. And I can tell you, when we first started genetic screening, it was really only BRCA1 and 2. Now we're up to screening for 50 genes. I think soon we'll be up to screening for well over 100 genes. So that's where I see us really making a difference in terms of being able to categorize risk, is identifying the risk associated with these pathologic variants. Now we know there are so many, like RAD51, and, you know, that confer very significant risk that we didn't know before. ATM, you know, these were genes that 10 years ago, I mean, we didn't even know what they were. Now we know that they can confer very significant risk for breast cancer. And we're finding more of those, you know, every month.
Kristen, did you have any questions?
Yes. And I know there was one question about developments in vaccines for early stage breast cancer. I feel like maybe we answered that sufficiently.
Yeah, the vaccines for early stage breast cancer, very definitely. The problem with early stage breast cancer is to show that you prevented disease recurrence, you would need 1,000 patients in 25 years. So one of the things we do want to work with is the FDA. If we can identify, let's say, that type 1 T cells really are a surrogate of clinical response, what you want to do is find an approval that doesn't require so long of a time. So a lot of our clinical trials as we move forward are really focusing on, like for the COVID vaccine, that we know that you need a certain level of antibodies to protect against COVID. What we're trying to identify is what is that level of type 1 T cell we need to hit, that magic number that says the vaccine's going to work. Then you can rapidly develop vaccines and get them approved based on that. So to get into early stage cancer, we really need the help of the FDA and experts to help us figure out a faster development pathway.
And for STEMVAC, I know your trials are broken up into, you weren't doing HER2 positive, but will there be a vaccine, in your opinion, that covers all of the cancer stem cells? Are they that similar, or does it need to be something for HER2 positive, something for ER/PR positive, and something for triple negative?
I think right now we're focused on the subtypes, something for HER2, something for triple negative, something for ER positive. But you can almost envision you go in and you get your HER2 new vaccine, and then you get a booster shot with the STEMVAC vaccine. So there are regimens that we can come up with of vaccination that can give you kind of a more complete picture. I think immunizing against metastases is another vaccine that we have under development, where the data look very, very good in animal models. And that might be like a second vaccine regimen once you finish the vaccine that is specifically targeting your disease, your subtype of breast cancer, then to go on and get an anti-metastatic vaccine.
Does that work for the patients that have already, like myself, who've been diagnosed metastatic and have been in remission for a long time?
We have not yet tested that in animal models, but that's something that we want to do.
Sounds promising.
How difficult is it to take a trial, or what are the biggest challenges, I guess, from the mouse model into humans? Because it doesn't always work.
Right. And, that's the thing. The development pathway is very long. So you can develop a vaccine and test it in animal models. You can come up with a clinical trial idea, but then you have to go through the FDA, which is a very long process. And you also have to manufacture the vaccine, which is a very expensive process. Then once you get into clinical trials, the faster you can enroll the trial and get your answer, the faster you can figure out whether a vaccine works. So the animal data really provides you some assurance that there's activity. Would I take forward a vaccine that had no effect in a biologically relevant animal model for the disease? No. Why would you? If you have no hint at all that it would have any applicability to humans. But you're right. Some vaccines just don't work. Our concept for the collaborative is anyone who's got a good idea, and there's a lot of people developing breast cancer vaccines. It's a very hot area of research. You can read in the paper almost every week, there's some report of a breast cancer vaccine. Get those vaccines, bring them all together in a network. Everyone open the vaccine trial at their place so that all patients have access to it all over the country. And we enroll that vaccine trial in 3 months. We analyze the vaccine trials all the same way on agreed upon parameters, type 1 T cells. And those that generate the highest levels of type 1 T cells go into clinical trials. Now, what happens is you have research groups that develop a vaccine. It's taking them decades. The vaccine doesn't really work very well. It's not very immunogenic, but they keep pushing it forward, right? Because they're working in a silo and they see the trees instead of the forest. I think by working as a large group of scientists together, looking at all the other data and making decisions, people can say, no need to push this forward. We need a sea change. Or maybe I should take my antigens and analyze them the way Nora does. Let me go back to the lab. So we can all learn from each other. You know, I'll see someone who has high levels of immune responses generated with the same assays that I use, and they have some unique thing going on in their vaccine. Let me add that to my vaccine and see. So this is how we just build on the knowledge. Everyone looks at everyone else's data. We do shared decision making about what looks more promising, and we commit to running those clinical trials as quickly as we can. That's where we make progress. So, like your question, you know, I have stage 4 disease. Can I get the vaccine? And the fact that we ran this clinical trial and it's going to be another 2 years before we open the clinical trial that you would qualify for. Why does it take that long of a time? Why can't that trial be opened right now? That's what we're trying to do with the collaborative.
And is a successful trial for you and I guess for the FDA ultimately one that produces an immune response, or are we waiting to see lack of recurrence?
It has to be lack of recurrence. It has to result in overall survival. That's why the more advanced stage patients are really where we're trying to test, because we'd be able to get the answer in a shorter period of time. Then, if we can really do all the laboratory analysis, and we've got very quantitative, specific tests, and demonstrate statistically that this correlated with the outcome, that's where you begin to develop a surrogate for response. And that's what we desperately need in vaccines. That will allow us to go into early stage breast cancer.
Great. Well, that's all I had. I'll let Jillian say goodbye ultimately, but I just wanted to thank you. I'm so excited about the research you're doing and getting to work with you on this collaborative. I tear up when I listen to you talking about a vaccine and the possibility that it could come soon for us and for those we love. And so thank you so much. I hope people got a lot out of this. And again, we'll just be updating this community as our work moves forward with the collaborative, and we hope everyone will join us in this fight.
Thanks for the opportunity to share our data with you.
Yes, thank you, Dr. Disis. This has been wonderful and so informative and so much hope moving forward. I can't say how much this means to someone like myself who is just living every day, hoping to see the next. So thank you for that and all you do. I will share the recording of this with everybody who's registered. And if you have any follow-up questions, please don't hesitate to email me directly and I'll pass them on to Dr. Disis. Thank you, everyone.
Thanks, everyone.